We like to think of scientific classification as this really neat, orderly thing. You want clarity, precision, a place for everything.
Right. But the reality, especially when you get down to the level of subspecies, is that
Well, it's messy.
it's really messy, and it gets even more complicated when you're dealing with the kind of subtle variations you find in, say, the rainforests of Southeast Asia.
It's a profound challenge. I mean, historically, classification was often just subjective. A scientist looking at a museum specimen and basically saying, this one looks different enough.
Uh,
And that's led to subspecies being called, and this is a quote, the most critical and disorderly area of modern systematic theory.
Disorderly. And that disorder is just compounded by, frankly, neglect. The Oriental region, we're talking Malaysia, Indonesia, is one of the most neglected areas for this kind of research.
Absolutely. So our mission today is to really unpack a major effort to bring some order to that chaos. We're doing a deep dive into a study on the chestnut-winged babbler species complex.
That's Cynoderma erythropterum and C. bicolor. Exactly. And this group was a perfect test case because it was just a taxonomic quagmire.
A quagmire is right. Historically, you're looking at something like 10 different subspecies being described over time, but modern consensus had kind of whittled that down to just 5.
So the researchers knew they couldn't just trust their eyes anymore. They needed objective, quantitative methods.
And this is where it gets really innovative.
It is. They used this multi-pronged approach, combining old-school taxonomic rules with some pretty cutting-edge digital analysis. They started by digitizing the colors of the bird specimens.
Taking measurements of feather colors using models like RGB and HSL.
Yep.
Okay, let's unpack this. Because digitizing color sounds simple, but the way they did it to remove subjectivity is kind of revolutionary. They didn't just look at the raw numbers. They brought in an algorithm called CG2000.
And CG2000 is a total game changer because it moves way beyond just simple color subtraction. If you have 2 shades of brown, just subtracting their RGB values doesn't really capture how a bird or even a human perceives that difference.
Right.
The CIE2000 algorithm, it mathematically models human visual perception. It accounts for the weird, non-linear way our eyes register subtle shifts in color.
That's the key detail, isn't it? They're not just measuring light. They are quantifying the biological perception of color. That's a massive leap forward. It turns a vague description in an old field note into hard, usable data. So before we get to what they found, let's talk about the rules of the game. They decided to rigorously apply the classic 75% subspecies rule. Can you just remind us what that actually means?
Sure. The 75% rule is the long-established benchmark. It basically says that for a specific trait, in this case, color, 75% of the birds in one population must fall completely outside the range that contains 99% of the birds in the other population.
Wow. So it's a very high bar.
A very high bar. It's designed to prove that the two groups are overwhelmingly distinct.
And these rules, I mean, they're great in theory, but they're notoriously hard to apply in the real world, which is why people often just skip them.
They do. But these researchers applied them consistently. And they immediately ran into a huge technical problem with the color data itself.
What was that?
Well, when you measure color, you get a whole bunch of correlated parameters, hue, saturation value, red, green, blue. It's a lot. If you just look at, say, redness, the signal is incredibly noisy because it's influenced by everything else.
That sounds like a massive hurdle. So how do you get from a dozen messy, overlapping readings to one single useful number? This is where they had to linearize the data, right?
Exactly. They used a combination of principal component analysis, PCA, and crucially, linear discriminant analysis, or LDA.
Right.
Think about it like this. PCA finds the main sources of variation, but LDA takes that and statistically twists it to maximize the separation between the groups you think are different.
So if they think one group has a darker breast, the LDA process basically squishes all that complexity, hue, saturation, the works, into one single number. They call it LD1. And that number perfectly captures how dark is this breast in a way that makes the two groups look as different as possible.
Precisely. LD1 becomes your ideal measurement. So with that tool, they first tried an even stricter rule, the 95% subspecies rule, which some modern taxonomists prefer.
And how did that go, the stricter 95% rule?
It failed spectacularly.
Really?
Yeah. It couldn't even diagnose pairs that were, visually obvious to anyone, like the clearly darker breast of the Pyrrhophium subspecies compared to Erythroptera.
Wait, hold on. You're saying they had two birds that any expert would look at and say, yep, those are different, but the objective 95% rule said, nope, not statistically distinct.
That's exactly right. And that's a huge insight for taxonomy as a whole. It really suggests that the 95% threshold might just be too strict for this kind of subtle divergence, and it could actually be hiding real valid boundaries.
So the 75% rule was the sweet spot.
It was.
But you mentioned that rule requires big sample sizes. What about the rare subspecies, the ones from a tiny island where you might only have a handful of specimens in a museum drawer?
And that's where the CIE 2000 algorithm came back in for a second trick. They couldn't use the 75% rule on, say, the USNM collection for some of these birds, but they could use CIE 2000 to measure the precise pairwise color distance between every single specimen.
So instead of population overlap, they just measured the literal distance between two colors.
Exactly. And from those distances, they generated these branching diagrams, these phylograms. And those diagrams just visually showed how specimens clustered based on color alone. It was a perfect workaround for small sample sizes.
So with all those tools in place, let's look at what they actually found. When they started measuring different parts of the babblers, which traits actually mattered?
Well, the results were a bit surprising. Mantle coloration, the color of the bird's back, turned out to be a pretty poor diagnostic tool. Most of the subspecies pairings failed the 75% rule on back color. It just wasn't consistent enough.
So the back color is basically just noise. What about the front, the breast?
The breast coloration gave them much better resolution. Using the 75% rule, they could reliably diagnose 3 key distinctions, all involving the subspecies Pyrophium.
Okay.
It was statistically confirmed to have a darker, more distinct gray breast compared to its neighbors.
So breast color helps sort out the continental birds. But the really definitive feature, the one trait that gave them a clean break across the whole species complex, that was on the head.
The crown color angle. This is the most elegant and just clear-cut result of the whole study. They measured the angle at which the chestnut color from the back reaches up onto the crown. And this one feature gave them a 100% distinction between the major groups.
100%. So what did that simple angle show?
For the continental group, C. erythropterum, from the Timali Peninsula and Sumatra, the angle was consistently 0 degrees.
0 degrees, meaning what?
It means the chestnut color goes all the way to a point right above the eye. They have a full, rich chestnut crown, like a full hat.
And the Bornean birds, C. bicolor, the ones with the gray crowns.
They all had non-zero angles, averaging around 64 degrees. So the chestnut stops way short, leaving the crown mostly gray. And that one measurement just perfectly splits the 2 core species. No ambiguity.
Which would be absolutely crucial when they started looking at those weird intermediate island forms. Right, let's talk about the consequences. Because this is where the hard numbers led to some real drama in the taxonomy world. It challenged some long-held assumptions, and it even resurrected a subspecies everyone had forgotten about, C.E. apega.
That's right, C.E. apega. It comes from the Banka and Belitung Islands, and for years it had just been synonym lumped in with its neighbor Pyrophium. But the quantitative analysis just screamed that it was distinct.
What made it so distinct on the measurement table?
Well, morphologically, it was perfectly intermediate. Its neighbor Pyrophium has that full chestnut crown, that 0-degree angle. Apega, though, had a partially slate-gray crown. Its angle is about 38 degrees. And the CIE 2000 data also showed its mantle color was statistically distinct.
So it's physically in between the full chestnut group and the gray-crowned group, which creates this huge question mark. Is it a weird subspecies of Erythropterum, or is it maybe closer to the Bornean species, bicolor?
Exactly, especially since those islands have known faunal links to Borneo. The color data was ambiguous on species placement, but it was crystal clear that the population was unique.
So they need a tiebreaker.
They turned to bioacoustics, the bird songs. In babblers, vocalizations are often really fixed reproductive barriers.
And what did they measure in the song?
They measured the ratio of the first pause between notes to the second pause. For the apega specimen, the recording gave a ratio of 1.07, and that number fell squarely within the known range for C. erythropterum.
So its voice belonged to the continental group.
Unmistakably.
That is just such an elegant solution. The color data proved it was unique, and the sound data placed it on the family tree, a resurrected subspecies.
But then on the flip side, the data also acted as a pruning tool. The researchers ended up advocating for synonymizing a subspecies that is, right now, widely recognized, C.E. fulviventra from the Banyak Islands.
So getting rid of an accepted subspecies, that's a big step. What did the data show there?
When they used the CIE 2000 method to create those phylograms, fulviventra just failed to cluster distinctly. It didn't reliably separate from pyrophium or another form called pelum.
And pelum was already thought to be a synonym of pyrophium.
Right, so the conclusion was that the differences people had noted for fulviventra were really just subtle variations within one single, broader subspecies.
You lump them all together.
Lump them. It just perfectly shows how these methods can both expand the tree where we've overlooked things and prune it where we've been a little too eager to split hairs.
Okay, let's pivot to Borneo now, home of sea bicolor. There was this big uncertainty about the boundary between the northern subspecies, bicolor, and the southern one, rufum. How did that crown color angle help clear up that map?
Well, CB rufum is supposed to have a more rufous color that extends a little further up the nape. By applying the objective angle measurement, the researchers found that the range for rufum had been severely overestimated.
So the boundary line on the map needs to move. A lot.
A lot further south, yes. They could only find one specimen that was unequivocally rufum, collected way down near Banjarmasin. It had a lower crown angle. The data strongly suggests that rufum is highly restricted, probably just to the flat, swampy central Kalimantan Depression.
And the rest of the island, even the far south, is actually CB bicolor.
Correct. But then, the research took this really unexpected turn. Within that widespread bicolor population, they found something called a morphological cline, a gradient of change across the geography.
A highly significant north-south color cline.
Yes. As they tracked specimens from the south of Borneo all the way up to Sabah in the north, they found this really strong correlation between latitude and the crown color angle.
Tell us about that. How does the color actually change as you go from south to north?
The angle of the color change steadily increases. In the south, the average angle was about 59 degrees, so the chestnut reached a little further up the head. But in Sabah, in the far north, the average angle was almost 68 degrees.
Which means the birds in the north have the most extensive gray on their crowns.
Exactly. The least amount of chestnut.
So if we connect this to the bigger picture, usually you'd expect a color cline to be driven by natural selection, right? Following something like Gloger's rule, darker birds in more humid places.
That's the textbook explanation. But the thing is, Borneo is one of the world's largest, most uniform rainforest environments. The whole island is humid. If this was just about adapting to climate, you'd expect a much less pronounced pattern, or maybe even none at all.
So if it isn't the environment, what's driving this subtle but measurable color gradient?
The researchers hypothesized that because the environment is so uniform, this cline is likely being driven by something else. Sexual selection. The color could be a visual signal, something used for mate choice or for recognizing your local population. A subtle marker of reproductive isolation that's happening right now.
And that is a massive insight, that these tiny morphological differences might actually be hidden social signals. This whole deep dive it really underscores the impact of applying this kind of rigorous quantitative analysis. It proves that just relying on subjective visual cues, it leads to misidentification. It leads to disorder.
And so what does this all mean at the end of the day? The study gives us a dramatically streamlined, scientifically validated taxonomy for these babblers. They basically consolidated a bunch of old names into just two main groups, C. erythroperum and C. pyrophium.
And, of course, big payoffs. The resurrection of C. apega, brought back from the dead by digital color and confirmed by sound. And then redrawing the map to restrict CB rufum to that tiny swampy bit of southern Borneo.
And what's really fascinating here is that the study validates plumage color, when you quantify it this way, as a legitimate tool for delimiting subspecies. It can stand right alongside genomics and bioacoustics. And that is such essential work, because if we're going to fix that disorderly area of modern systematic theory, we need tools like this. It's critical for conservation. You have to know what you're trying to save.
Absolutely. And it leaves you with this really provocative thought, especially about that Bornean color cline. If that gradient in crown color really is being driven by sexual selection, a social factor, and not just the environment, it begs the question, how many other subtle differences we see in tropical species, things we've always just dismissed as trivial geographic variation, are actually crucial hidden signals of divergence? Signals we can only see if we measure them precisely. We could have thousands of cryptic species out there just waiting to be uncovered, all because we're relying on the naked human eye instead of the CCE 2000 algorithm.